At this time, I would like to just provide a land acknowledgement. Scugog is situated on treaty land that's steeped in rich indigenous history. Today, we acknowledge that we are located on the traditional territories of the Mississaugas of Scugog Island First Nation. We recognize and deeply appreciate the historic indigenous connection to this land and our watershed, and recognize the contributions First Nations, Métis, and Inuit peoples have made, both in shaping and strengthening this community in particular, our province and country as a whole. We're grateful for the opportunity to meet here virtually, and we thank all the generations of people who have taken care of our land for thousands of years. Well, thank you. I'd like to particularly welcome representatives from Township Council. I've seen Mayor Watton and Regional Councillor McDougall, Councillor Coyne. I think we're also expecting Councillor Leroy. And I see several members of our Scugog Environmental Advisory Committee. I think Keiko Louie and Steve Ardren and Mr. Pinto are with us. I think we're expecting Danielle from Kawartha Conservation to join us as well. So thanks to you folks, and also to everyone else who has joined us. A really, really important topic. You know, it's for those of us who deal with Phragmites, we know it's a tough, fast-spreading, pervasive, just dense, invasive grass species that's really, really threatening our sensitive habitats, particularly wetlands and riparian areas. I have a swath about, I'd say, 1,000 square feet down on my road beside our driveway. And I've been trying to deal with that for about 3 years. It's kind of interesting in our case because my wife and I run a native plant nursery at the top of the hill. And so when people come in, I don't want them to see Phragmites in a situation where it's potentially going to impact our habitat. We don't use pesticides, so we have to deal with it manually, but really interested in hearing about the topic tonight. With that, I'm going to ask Barbara Cartine, who's our Director of Research, to introduce our guest speaker. Barbara.
Thank you very much, Rob. I too have a great swath of Phragmites running all along the front of my property on Reach Street. So yes, this is a big thing for me too. But for the lake stewards, it's vitally important to protect our Scugog watershed wetlands from both development, interference, and invasive species. Healthy wetlands and shorelines are vital to the health of our lake and our community. Even though we are so close to the GTA, we have a large diversity of interesting animals, birds, and fish that enjoy our 172 kilometers of lake shoreline and our 22.2 square kilometers of wetlands, all of which are vulnerable to negative effects of this invasive plant that Dr. Jones will be discussing. Since invasive plant species cannot be treated by traditional control methods, we will all be very interested to hear about Dr. Jones' research into a possible biocontrol. Dr. Ian Jones is a research associate at the University of Toronto in the Smith Forest Health Lab. His research focuses on insect, and insect-plant interactions and the ecology of introduced species. Ian received a bachelor's degree in biology from the University of Birmingham in the UK, a master's degree in applied entomology from Imperial College London, UK, and completed his PhD at Florida International University. Ian's current focus is on the classical biology control of invasive plants. Through an understanding of the ecology of biological control insects and their interactions with native species, Ian seeks to improve the efficacy, and safety of these beneficial organisms, contributing to more effective and sustainable control of invasive weeds. There you go, Ian. It's all over to you.
Perfect. Thank you very much. Let me do the lengthy sharing process. Let's see how quickly I can get this done. There we go. How's that? Can people see that?
You did that very quickly.
Yes.
Yes.
Perfect. All right. Well, yes, so thank you very much for having me here today. This is kind of a great opportunity to kind of get the word about biological control out there, but also to kind of learn more and just be part of the community that's got this same goal in mind of controlling Phragmites. So kind of a tiny bit more about me. After my PhD in Florida, I've done two postdocs. One was at the USDA in Florida, and I was working on biological control of invasive weeds there for just over a year. And then in 2017, I took a postdoc at the University of Toronto. So I've been in Ontario since 2017 and working on kind of various, some of the biggest kind of most problematic weeds that we have here in Ontario. So Japanese knotweed, dog strangling vine, and now most recently Phragmites. And I do think that kind of all of those weeds, Phragmites strikes me as the kind of the biggest and most immediate problem. And hopefully there'll be some kind of good news coming out of this talk in that we do have, I think, what looks like it should be a really effective biological control program for this plant. So it's a question of over the coming years seeing how effective that might be. But for now, we're in the process of just trying to implement it. So that's what I'm going to talk about today. It's the implementation of this biological control program on Phragmites in Ontario. So kind of to give a little overview of what I might cover. So first of all, I'll talk about Phragmites and define the problem. I won't spend too much time on that because I know you're all too familiar with the problems surrounding Phragmites. But then I'll talk a bit about what classical biological control is, and particularly how we make it safe, how it's different from the kind of disastrous biological control programs of the past, like introducing cane toads. I'll then talk about the specific agents that we're releasing in Ontario. That's Arcanara neurica and Lanisa geminipuncta, and just give a kind of broad overview of how the biological control program's going, like how we release the insects, how many we've released, and how they seem to be performing in the field up to now. So then I'll talk about two little experimental pieces. So as we kind of implement this biological control program, there's always a continuous research program going on alongside it. And that's to try and improve the kind of the efficacy of the system, how quickly we can get those insects established on the landscape, and also to ensure the safety. There's obviously continuous monitoring of the insects for that reason. And the two things I'll talk about largely are kind of work we've done to show that we can conduct releases of these insects across the kind of broad range of hydrological conditions that Phragmites grows across. And secondly, just a little bit about some work we've started on the use of drones for monitoring Phragmites, but more importantly for us, monitoring the activity of the biological control agents. So then I'll finish by talking about two kind of new directions for the program. These are the areas we're looking to expand. And one is redistributing insects from nurse sites, as we call them. So all of our biological control insects that we've released so far have been reared in the lab. So they're mass reared insects in a lab. We then take out and release in the field. But now that we have populations established in the field, we can actually come up with methods to just transport insects from one site to another. And that's going to massively increase the rate at which we can release these insects and get them spread across the landscape. The final thing I'll talk about is our hope to integrate biological control with other successful conventional management tools for Phragmites, things like chemical sprays and manual removal. These are things that can be very effective in certain situations, and it's just a question of seeing where biological control fits in that kind of that matrix of different control methods. I think it's clear to me certainly that if we're going to tackle Phragmites effectively as a community, it's going to need all of these tools, and it's going to need collaboration between all of these people. And so that's what we're hoping to create. So what's the problem with Phragmites? Well, this picture is probably as good as any to define that problem. Phragmites grows in these huge monocultures that just leave absolutely no space for other native organisms. And so when you get these massive unencumbered monocultures of Phragmites, you lose, first of all, many native species. And it's the plant species that tend to be the first to go. So all this diversity of plant life that we would see in wetland habitats and prairie habitats, like blue joint grass and Canadian milk vetch, they're completely eliminated in many cases from these environments. And the loss of those plants then has a knock-on effect on fauna in the area as well. So one example of that is nesting birds. So in Phragmites beds, we see a huge reduction in the numbers of nesting birds compared to reed beds made up of native species. And that's because birds like mallards and the least bitten, they use spaces in between these reed beds where there are small openings, and they then kind of curve the reeds over the top of the nest to create this kind of shelter. In a Phragmites bed, each year the stems remain standing from the previous year, and even sometimes from 2 years before, and you get these incredibly dense beds of almost kind of woody bamboo-like material, and there's just no space and material for these birds to make their nests. And a similar one is the marsh wren, slightly different in that marsh wrens create these kind of really beautiful, kind of ornate globe-shaped nests. But in a Phragmites reed bed, they just don't find the materials they need, the kind of flexible and small materials they need to make those nests. And so it's another species that is not necessarily lost entirely, but exists in far lower numbers in these Phragmites infested areas. Fish are also affected. So this is a mumachog, usually around the kind of Spartina cordgrass that they live around as young fish. There will then be kind of gaps in that Spartina where there are kind of recesses in which these fish create nurseries for the young fish. Again, you look on the right-hand side of the slide there, and all of those recesses, all of those gaps in the reed beds are gone. And so those kind of safe nurseries for the young fish are completely eliminated, and we lose populations of these fish among other species. Turtles are another one that are badly affected. So these are the kind of 8 commonly seen turtles that are native to Ontario. And of these, the one that's been studied most closely in this context is the spiny softshell turtle. But many of them suffer the same problems. And that is that these turtles lay their eggs, they bury their eggs under the ground in the spring, and they need those nest sites to receive a decent amount of sunlight to keep the nests warm enough for the embryos to develop. But in the case of Phragmites infestations, what tends to happen is that they lay those eggs in the spring, and within a very short space of time after they've laid those eggs, the landscape around those nests changes completely because the Phragmites stems have grown up to 3 or 4 meters tall and largely shading out those nests. And so many of them are lost, and the kind of viability of those eggs are hugely reduced. And so this is another species where we see big impacts from the Phragmites. It should be said that all of these kind of ecological impacts, and these are the things that kind of get me interested, but along with that, you have kind of impacts on infrastructure and even safety in many cases. So you have Phragmites growing along roadsides, which we're very familiar with seeing driving around southern Ontario. But without treatment, those Phragmites populations will kind of start to encroach on signs, making kind of safety signs unreadable, and even affect sight lines on roads. So this is a major safety issue. And then there's the image on the right shows these are culverts that are required for drainage from areas. And the water flow along those culverts is just being completely blocked in many cases by these Phragmites infestations. So these are kind of municipal issues, and they're issues that tend to be needed to be addressed immediately. So not necessarily a job for biological control in this case, but just another part of the kind of the issue surrounding Phragmites. So we do have some good methods, some effective methods for controlling Phragmites. So in kind of relatively small but kind of problematic areas of Phragmites, we can use things like spading. So this is where we just take a spade and cut the Phragmites stems an inch or so below the soil level. And in so doing, we can kind of drown those stems and remove that patch of Phragmites. And it can be very effective. And we have lots of community groups doing this that do an amazing job of kind of reducing the problem over time. We have chemical control, which works brilliantly well in many situations. We have situations like in Long Point, where there's been a kind of long campaign to spray gradually many of the Phragmites patches around that area. And they've done a brilliant job in kind of removing the Phragmites there. We're now starting to see some remnants of Phragmites returning in that site, but much slower than we might have thought. There probably will need to be additional chemical sprays, or maybe biological control might be an option to kind of to come in there and help damp down that return of the Phragmites. There are other methods as well. So burning is one. This is usually done in the winter, and it doesn't actually do much to kill the Phragmites. But what it does do is it removes that kind of excess of above ground biomass that allows more sunlight to get to the ground, which just gives some native plants a fighting chance of getting a foothold back in these environments. And we have these quite serious looking machines. This is a marsh master. So these can get into some infestations and cut the Phragmites, but also mulch it at the same time. So again, similar to burning, it does a good job of kind of removing all of that biomass and kind of pressing reset a little bit on that site. But what it won't do really is damage the rhizome underneath the ground of the Phragmites. And so we will inevitably see Phragmites return. And so all of these conventional control methods, as useful as they are and as necessary as they'll be, there are limitations. And those limitations really come in the form of kind of scalability. This is an infestation that's province-wide, and it costs money to use a marsh master and to spray herbicides. So we need to get the kind of the most sensitive areas, but there's a need for a kind of scalable and sustainable tool that we can use in conjunction with all of these things. And that's what biological control is. So I'll talk a little bit about what biological control is. And then, as I mentioned, I'll talk more about how we in modern biological control ensure that it's safe. So this is kind of the major explanation of what classical biological control is. Here you have Phragmites growing in Europe in its native range. And in Europe, Phragmites grows in conjunction with a whole suite of herbivores and natural enemies that knock it back and reduce its fitness such that it kind of plays much more nicely with other native plants in Europe. So if you go and see a patch of Phragmites in Europe, you'd hardly recognize it from the Phragmites that we're seeing here in North America, because it just grows much more sparsely in and amongst other native vegetation. When Phragmites makes the move over to North America, in the absence of any of its natural enemies and herbivores, it now just grows unencumbered. And so its fitness is through the roof, its competitive ability is through the roof, and we see these kind of huge monocultures of Phragmites taking over everything else. So the idea of classical biological control is that we reunite the Phragmites with one or more of its natural enemies or herbivores. So the hope is that having kind of established these herbivores on the landscape, they will do enough damage to the Phragmites just to bring it back so that it's growing more like it would grow in its native range and playing more nicely with those other native plants. And so that's an important point to raise, is that the goal of biological control is not eradication of Phragmites. That's not a realistic goal for us at this point, for any control method, quite honestly, but certainly not for biological control. The goal of biological control is to damage the Phragmites just to an extent that it exists with other native plants, and it exists below a threshold whereby it's not having ecological and economic negative impacts on the region. So a kind of similar story to kind of bring your attention to, and many of you may be more than familiar with this, and that's the story of purple loosestrife. So these photos aren't brilliant, to be honest, but on the left-hand side, you can see a kind of much more purple hue to the wetland there. And that's because at that time, 20 or so years ago, this area of the wetland was almost completely dominated by purple loosestrife. This was another invasive plant that was taking over and growing in this kind of invasive habit. After the introduction of 2 biological control agents, the second photo is taken 10 years later. And although you might see the odd sprout of purple in there, the community has changed massively to incorporate many of the native plants again. And so this is the kind of story that we'd hope to repeat with Phragmites through biological control. So I mentioned I'd talk about the safety of biological control. This diagram you can see at the top of the slide here is the different phases of a biological control program. And it starts with something we call horizon scanning or foresight. And that's essentially looking at Phragmites and saying kind of, what's the ecology of this plant? Is this a situation where biological control would be useful or is necessary? Once we've established that that's the case, we move on to foreign exploration for potential biological control agents. So and I used we loosely, this was nearly 30 years ago. So colleagues from way before me went back to Switzerland to look for potential insects that could act as biological control agents for Phragmites. So this was happening in the late 90s. Within a couple of years, they'd found a couple of insects that they felt were not only fed on Phragmites and did significant damage to the plant, but that were completely specific to the plant and wouldn't damage anything else. So that's all we're looking for, even when we're looking at the candidate stage for agents. But once we've identified those insects, we then enter the risk assessment phase. And that's the longest and most robust and rigorous phase of any classical biological control program. And it takes the form essentially of host range testing. So we use this method called the centrifugal phylogenetic method. What that means is we're taking these candidate insects and we're exposing them to a huge list of plants that we know they may come into contact with in their introduced range. And we start with the plants that are closest related to the target weed. So in this case, we would be introducing them to our native Phragmites from here in Ontario. And then we would be working with plants more and more distantly related until even we get to plants that are not very closely related at all, but that might have specific economic or cultural significance. So maybe crop plants and things like that. So the list of plants will be around kind of 60 or 70 plants. With each plant, we're testing them individually to see, will these insects feed on those plants at all? Will they lay any eggs on those plants? Are they capable of completing any part of their life cycle on those plants? If they're feeding on the Phragmites, but the Phragmites is taken away, will they then switch over onto any of those plants? And these tests for the two agents we're working with on Phragmites went on for 11 years. So this is a really kind of rigorous process. And those agents, as I mentioned, Arcanara neurica and Lanisa geminipuncta, were eventually petitioned for release only in the kind of mid-2000s. And so since that time, we write the petition. It goes through a long process of being assessed by different bodies. And ultimately, in 2019, we got the green light to release these two insects in Canada. So by this time, the project is at least 30 years old. So this is a kind of an important thing to bring up, I think, because the natural question, I think, for anyone who's kind of comes across the idea of biological control is to say, well, is it safe to release these insects? And I think, you know, especially when you have examples from the past, kind of releasing cane toads in the 1970s, these are things that will kind of bring up question marks and make people concerned. And that's obviously completely understandable and normal. I think ever since this kind of host range testing has taken place, since this has been the norm in classical weed biological control, there have been no cases of non-target effects of classical weed biological control agents. So we're really happy with the level of safety of these insects. But the main thing is also to say that as classical biological control practitioners, those questions that you ask us about the safety of the insects, those are the questions that we spend most of our time asking as well. So we're certainly with you on that one. So here are those two agents that we've been releasing. It's Arcanara neurica and Lanisa geminipuncta. They're two noctuid moths, pretty similar in size and appearance, and they're about the size of your thumbnail as adults. But the key thing about them is that as larvae, they feed internally on the Phragmites stems. So the eggs will hatch and the larva will enter the stem, feed in that stem, damaging it, and then move on to another one. And so that's good because they do intense damage to the stems, but they're also pretty impervious to predators in this introduced range. We don't see many of them being eaten by birds or anything like that because they're inside those Phragmites stems. So this is what their life cycle looks like. They just have one generation per year. So the eggs are laid and they hatch in the spring. And then these larvae go through 5 different instars or larval stages, each one of them feeding inside a different Phragmites stem. So during the development of a single moth, that moth can damage 4 or 5 Phragmites stems. Then when the larvae are fully developed, they pupate in a different Phragmites stem, which they will also damage to some extent. And then the adults emerge to fly in kind of late July and August. So the females will lay about 150 eggs underneath the leaf sheaths of those Phragmites stems. And it's underneath those leaf sheaths that the eggs remain for winter. So these eggs are the overwintering phase. So when you look at patches of Phragmites when you're driving along, it's possible that underneath those leaf sheaths that you can see on the stems, there are eggs for these biological control agents. Now, the kind of overwintering and the position of those eggs is relevant for 2 reasons. One, because it plays a major role in some of the ways that we're looking to transport insects from one site to another. But secondly, it's another additional tool making these insects specialized on this invasive Phragmites rather than the native Phragmites here in Ontario. And that's because native Phragmites, if you're familiar with it, during the winter, it sheds all of its leaf sheaths. And so if you look at it at this time of year, the stems are completely smooth, almost like kind of very narrow bamboo. So if there were any eggs erroneously laid underneath the leaf sheaths of native Phragmites, those eggs would be lost and would die once those leaf sheaths are shed in the winter. So it's a nice tool kind of further keeping these insects apart from the native Phragmites. So we've released now 30,000 of these insects across the province, and we release them in 2 main ways. The first is as eggs. So as I mentioned before, all of the insects we've released so far have been lab-reared insects, reared either in Switzerland or at the University of Toronto. And what we end up with is these Petri dishes full of eggs that you can see on the left-hand side. And we place around 100 of those eggs inside the internal cup that you can see in the middle of this release unit. When those eggs hatch, the larvae can crawl out of that internal cup and then drop through the mesh at the bottom of the main release cup. And then from there, they'll move around to feed on the surrounding Phragmites. But what's useful about these egg releases is that because those eggs are the overwintering life stage, we can set these releases up anytime during the winter or early spring, as long as they're set up before those eggs hatch. And so what that means is with a relatively small group of people going out conducting these releases, we can get around to lots of release sites over a kind of larger geographic area. And this is the method that we use to release the majority of the insects that we release. The other method is to release larvae. So this is a more effective release method, but much more labor intensive. So in this case, we let the eggs hatch in the lab, then we collect Phragmites stems from the field, which at this time of year are pretty small. We bring them into the lab and we cut them in the internode so you can see the gap within the internode. And using a paintbrush, we put a single caterpillar into each of those stems. And then we place the stems in a kind of florist foam block. So it's like a little bouquet of Phragmites stems. So those inoculated caterpillars will feed within those stems for about 10 days, after which they'll bore out of those stems and then search for surrounding Phragmites to feed on. So once we've set up one of these stem blocks, we've got about 10 days to get it out in the field for a release. As I say, this is a much more labor-intensive way of doing things, but we tend to release about 3,000 insects a year this way. So when we release these insects, we monitor them at those release sites twice during the year of release. And the first monitoring session happens about 4 to 6 weeks after the release itself. And we're looking for the amount of damage that these larvae have caused to the surrounding Phragmites. Because the larvae don't travel very far, all of the damage is pretty centralized around the release point. So we use these kind of 65 centimeter quadrats that you can see around the release point here. and we count the number of damaged stems within that quadrat. And the damage we're looking for is these characteristic boreholes that you can see in the central picture here. So if you see that on a Phragmites stem, that's one of our biological control agents. There's nothing that looks like that that any other herbivores inflict on introduced Phragmites. It looks like it would be difficult to find those boreholes, and it can be. You do get your eye in, but there are also other methods that kind of bring you to the stems where they are. So if you see branching on a Phragmites stem at this kind of time of year, it's likely that that's a response from the stem from damage from these insects. So if you keep looking at that stem, you'll often find a borehole. And the same is true of wilting at the top of a Phragmites stem, certainly at this time of year. The second time we monitor each release site in the year of release is in the fall. So this is not so much looking at damage within the stems, but looking at the overall fitness of the Phragmites and how that's been affected by the biological control agents. So we use this same 65 centimeter quadrat around the release point, and we compare that to control plots away from where the biological control agents were introduced. And we're looking at kind of things like fitness metrics for the Phragmites, like stem density, average stem height, panicle or flower formation, things like this. What we found pretty consistently across all of our release sites is that in these biological control release plots, we do see reductions in stem density, we see reductions in average stem height, and even more consistently than that, we see reductions in panicle formation, so reduction in the number of panicles that are formed. So this is kind of very preliminary impact data in terms of it's just at the scale of the release plot, and it's only in the year of release. But it's still very encouraging to see the damage that these agents can do and the effect that it has on the Phragmites. And we hope that will continue to happen and expand to a population scale. Some of our early test release sites, where we now have really big populations of these moths, we have one site in Aurora where there are basically no panicles being produced by this Phragmites at all. So you can tell the Phragmites has become really stressed by the presence of these moths and the damage that they're causing. And that's exactly what we want to see. We don't really have the data to display for that yet, but we're hopeful in the next few years we'll start to see that really encouraging stuff come out. So with each of our release sites, we try to get back to the release site every year after the release as well. And that's to see kind of whether the insects have survived themselves, whether they overwinter successfully. But it's complicated because within a second year, there's been an adult flight period. And so any damage to the Phragmites is no longer going to be centralized around that kind of release point. It could be anywhere within the Phragmites patch. And so we use a different monitoring method at that point. What we do is we run 4-meter transects all the way around the Phragmites patch, and we run time searches looking for damaged stems on those transects. And from that, we generate a value of damaged stems per meter squared for the whole Phragmites patch. This is what some of our kind of longer term monitoring data looks like. I realize this is a bit kind of much to take in over a small period of time, but the main things to take away really are that blue column there is the damage in the year of release. And then the following green columns are damage to the Phragmites in subsequent years. And what it shows is that in almost 100% of cases where we see damage in the year of release, we also see damage to the Phragmites the following year and the year after that. And so it shows us these insects are overwintering successfully in Ontario. They're reproducing on their own. And what we're also seeing in some of the sites where we've been able to monitor for 3 or 4 years, we see a gradual increase in the level of damage year on year. And that's exactly what we want to see, this kind of slow ramping up of the populations to a point that they start really impacting the Phragmites. So this is all brilliant news for this stage of a biological control program. So this is the kind of the big overview of the release program. This is our 50 release sites across the province. So we've started to get kind of fairly good coverage of releases across the province. Some of these are kind of small releases, some of them are big releases. And so we've still got a long way to go to get these insects out there across the landscape. But this is where we are now, having released 30,000 insects. Of those 50 release sites though, the vast majority of releases have taken place in really dry locations. And that's for reasons of accessibility, um, and reasons of safety in some cases. And we just don't know whether our release methods are going to work equally well across this range of hydrological environments where Phragmites grows. So we conducted an experiment at Lynn Shores Conservation Area at this patch of Phragmites here. So it's growing into an area called Cranberry Marsh. And there's this kind of leading edge of Phragmites in the marsh where there's about 30 centimeters of standing water. But it's a kind of linear patch, and on the other side of that patch is a kind of much drier habitat where the Phragmites continues to grow. And so what we did is, this is the study area at Cranberry Marsh, and we conducted 5 egg releases and 5 larval releases along both the wet edge of the Phragmites and the dry edge of the Phragmites, just to see which would perform better. So here's what those releases look like, the egg and larval releases above that standing water there. So you can imagine as the larvae drop out of the egg release cup or fall from the transferred stems, they're essentially going to be met with standing water that they have to navigate. So the question is that curtains for the larva at that point, or can they kind of make their way, whether it's kind of by chance or by their own motor skills, to get to a Phragmites stem to feed on. On the right-hand side, you can see the releases in the drier habitats. That's much more kind of akin to the kinds of releases that we've been conducting in the program before. So we monitored these releases in exactly the same way that we had previously, looking for those boreholes that you can see in the center. And the results look like this. So really encouraging. The first thing to say is that the larval releases were a lot more effective than the egg releases. That's completely normal. That's kind of exactly what we expect after the number of releases that we've done. But the other thing was that the releases in the wet environments did not do any worse. In fact, they performed slightly better than the releases in the dry environments. And so kind of what that does for us is it opens up a whole range of opportunities whereby we can continue to use our existing release protocols, but along a huge different range of hydrological conditions. And it could be that situations like at Lake Scugog, if there are patches of Phragmites that are kind of in deep water and that don't lend themselves to treatment with herbicides, for example, we now at least know that releasing biological control agents in those situations is viable and that it works. So we followed up that experiment looking at the effects of the Phragmites again, and we found that in terms of Phragmites stem density, the larval releases in wet environments significantly reduce stem density. And in terms of panicle formation, in dry environments, both the larval and the egg releases reduce panicle formation. And in the wet environments, again, it was only the larval releases that reduced panicle formation. Again, this is all kind of very preliminary stuff because it's in the year of the release, but it's always kind of encouraging to see situations where damage from the biological control agent directly leads to kind of measurable reductions in Phragmites fitness. Another quick thing to touch on is the use of drones to monitor Phragmites, but also to monitor the biological control agents that we're working with. This is a drone that we fly in conjunction with Ducks Unlimited Canada, and it has on it a multispectral camera. We fly it relatively low over several of our release sites, but one in particular where we have kind of experimental release points, and we have kind of very accurate GPS locations for those release points. So we can fly above that release area and we know where there are damaged stems. So we can then try and teach the drone to recognize when it's seeing damaged stems versus healthy stems. And there are certain kind of light metrics that you can use, like NDVI, that are effective at kind of recognizing damaged or stressed plants. And so those are the kind of metrics that we're working on to try and get these drones to be able to show us within a Phragmites population where our biological control agents might be active. So these tools can do a lot of things for us beyond that. So just kind of using these cameras to identify where the Phragmites is compared to native vegetation, potentially differentiating between invasive and native Phragmites, and again, identifying damage from the biological control agents. Because if we can do that, not only can we make an assessment of how effective they're being and how quickly they're dispersing, but it may also aid us in our ability to transport insects from one site to another if we know where to go within a site where they're very active to collect them and move them along. There's also the question of what's the best time of year to fly. And one of the things we're working on most recently is flying in the fall, because we now know that one of the biggest effects that these biological control agents have on the Phragmites is the reduction in productions of flowers or panicles. And it's very obvious, when it gets to the end of the year, you see kind of this sea of brown panicles on the Phragmites. And so it'll be very clear and obvious if we can kind of see patches where those panicles are absent or at least less dense. And this will give us an indication of where these agents have been effective. So I want to kind of finish now by talking a little bit about the new areas that we're looking to expand into as a biological control program. And the first is redistribution of agents from nurse sites. So I showed you those 50 release locations where we have the insects on a previous slide. And among those 50 sites, there's a smaller number where we released maybe several years ago.
Oh, oh no. Mm-mm-mm.
Hmm.
Hi, Rob. Can you hear me?
Yes, Mike, I can.
So we've just lost Ian then?
I think so.
Can we phone him? Maybe he's still speaking.
Well, we have his, Barbara might have his contact information.
Mm.
Mike, do you,
I don't believe I have his contact anywhere.
I don't see Barbara's name on what I'm looking at. Is she still on?
wonder if she's,
No, I don't see her on here.
that's too bad.
Yeah, sorry everyone. This is Heather. It looks like Ian looks like his Wi-Fi dropped or something, so I'm hoping he dials back in.
Yeah,
I just texted Barbara. I don't think she's on. She logged off to she would have his contact info, so I've
yeah,
asked Barbara to reach out. So why don't we just wait 5 more minutes?
yeah, I think if we can, just everybody can just
Hopefully
hold
he can
on.
log back
Oh,
in.
there he is. Hi, Ian.
Hello.
Can you hear us? We
Sorry about
lost
that.
you.
Yes, I'm sorry. My son unplugged the router to turn the air.
Oh.
Oh, no.
He's got to have his fish fingers.
Okay, can you set up where you were?
Yes, yeah. I'll let me.
I have to ask, how old is your son?
Yeah, he's 10.
Oh,
Okay.
So you
And
get
here we thought it was our technology.
he's off the hook, but
Okay, that's great to have you back, Ian. Thanks. Thanks everybody for waiting.
all right, let me I think this is where we were, but tell me if I need to go back further.
Yes.
Okay, let's see. The only thing is it's not now allowing me to move the slides forward. Oh, there we go.
Here we go. Good.
Yeah, perfect. So sorry about that. Yeah, so I had showed you previously a slide with these 50 release sites across southern Ontario. And of those 50 release sites, some of them we have kind of now really robust, kind of established populations of these biological control agents. And we expect to see, and we're monitoring to see, natural dispersal of these agents radiating away from these nurse sites into the surrounding landscape. But what we also want to do is artificially accelerate that process by actually moving insects from these nurse sites into new locations. And so the question becomes kind of what's the best way if you have a big population within an isolated patch of Phragmites, you have a big population of these biological control agents. What's the best way to move some of those agents into a new site and set up a new population there? And I mentioned before that these insects lay their eggs underneath the leaf sheaths of the Phragmites stems. And that's where the eggs stay from any time from kind of mid-August all the way through to late May when they hatch the following season. And so that to us is kind of, that's the life stage that we can kind of try and harvest. We have the most time and flexibility to try and harvest that life stage and then move it on to new sites. But the first questions we had to address were, is that feasible to do? If you go into a nurse site and you collect a certain number of stems, how many eggs are likely to be on those stems? And a second question was, if you can find those eggs on the stems, whereabouts on the stems are they likely to be? And so these stems that you can see on this slide, were collected from a nurse site in Waterloo. We collected 100 of those stems and then took them back to the lab and then very closely and carefully dissected each of those stems. And in total, from those 100 stems, we found 316 biological control agent eggs. So that for us was that's really, really effective way to harvest these eggs. And if anything, although it might sound kind of labor intensive to kind of painstakingly dissect big Phragmites stems like this, to generate 316 eggs that way was certainly no more work than it would take us to rear those moths in the lab. And so this kind of was a bit of an eye-opener that this is potentially a kind of seriously effective way of moving the insects around, even if we just dissect the eggs out of the stems and then release them in the normal way. What this graph on the bottom of the slide shows is what height on the Phragmites stems those eggs were actually found. And so the vast majority of them were found very low down on the stems. So what that means is if we want to kind of transport stems from one site to another to move the insects, we don't need to transport that whole 4-meter tall stem, particularly with a flower on the top of it. We can just transport the lowest 1 meter, and in doing that, we'll be transporting the vast majority of these eggs. So last summer, in the summer of 2024, for the first time, we trialed this process of transferring insects to new sites through stem transfers. So that was done at Ontario Tech University. These are some planters in the middle of Ontario Tech's campus. And these planters have been inundated with invasive Phragmites for many years. And so in the winter, we mowed that Phragmites, and then we transferred in 18 blocks of these stem transfers. So 75 stems in each case, collected from nurse sites, and then transferred into this site at Ontario Tech University. But in order to transfer those stems, we actually used 3 different treatments based on when the stems were collected from the nurse site and when they were how they were overwintered and when they were transferred into the Ontario Tech site. So the first group, the stems were collected in the fall from the nurse sites, not long after they were laid by the adult moths. And then they were transferred directly into the Ontario Tech site. So those stems and any eggs they contained overwintered in the Ontario Tech release site. The second treatment was stems that were collected in the fall, as before, but those stems were then stored in sheds at the University of Toronto for the winter, and then only transferred into the Ontario Tech site in the spring, shortly before the growth season. And then finally, the third treatment, stems were collected from nurse sites in the spring, shortly before the growth season, and then just transferred straight into Ontario Tech. So we had these stem transfers in place. As the summer progressed and the Phragmites grows up in these planters on the campus, we continue to monitor the surroundings of all 18 of those stem transfer treatment blocks. And what we find, happily, is that what we're looking for essentially is damage, that same characteristic borehole damage that you can see on the right-hand side. And what we found was significant amounts of damage in all 18 of those stem transfer treatment blocks. And so this showed us that this is a kind of really effective way of moving insects from one site to another. And so it opens up huge possibilities in terms of what we can do with our summer releases. What we also found was that those 3 treatments I described all worked equally well. There were no significant differences in those treatments. So what that means is that when it comes to conducting stem transfer treatments for us, we can collect stems from nurse sites whenever we can, anytime after the fall or through the winter. We can store them at University of Toronto, or we can put them straight out at release sites. We can wait till spring to release them. And all of these different combinations seem to work. And what that means is that because this method seems to be effective across this range of different criteria, it means it's more likely that we'll be able to kind of conduct more of these releases each year, and even eventually get to a stage where releases like this could be conducted not just by the kind of biological control, the small group of biological control practitioners that currently release insects, but could be done on a kind of community involvement basis. So the final point to raise is the new direction that we want to go in, which is integrating biological control with other conventional management tools. And so I've been on this Phragmites project for about 4 years. And during that time, and from long before then, we've had fantastic relationships with partners like Ducks Unlimited Canada, Nature Conservancy Canada, as well as many of the conservation authorities that we've been lucky to work with. And they've been absolutely kind of integral to the project in terms of providing us with patches of Phragmites where we could release our insects and access to those patches so we could monitor them and do our research. But the partners we've had up until now have not done an enormous amount of conventional control of Phragmites. And so we haven't had too many opportunities yet to release biological control in conjunction with these other management tools. In recent times, in this coming year, we've developed relationships with 407, and we'll be releasing at several sites along the 407 where there will also be chemical control taking place. So that's an opportunity for that. And we've also had discussions with the Long Point Biosphere region and several other regions where we may have an opportunity to do this. The way we see it is that the biological control program is in its infancy, and as positive as things look, we don't know exactly how effective it's going to be. And so biological control is in no way a replacement for any of these conventional control methods. Neither will it be if it's incredibly effective. It's just a tool in the toolbox that allows us to tackle some of the Phragmites where maybe these other conventional tools can't. And so what's nice for us is if there are situations where areas are being sprayed or they are having Phragmites manually removed, but there might remain some patches of Phragmites that are not logical to spray or not possible to remove. And that's where we imagine ourselves coming in, releasing biological control agents there. If we can establish a biological control agent population in those patches of Phragmites, then those agents should then be able to move out from that patch and start to pick off any Phragmites that grows back after the chemical sprays and things like that. So to summarize, we've released about 30,000 of these insects across 50 sites, and our monitoring is looking incredibly positive in terms of the ability of these insects to overwinter successfully and the damage that they do to the Phragmites. We now know, based on the kind of experimental work I described, that we have release methods that work across a wide range of hydrological conditions. And so that gives us opportunities, again, potentially in places like Lake Scugog, to treat areas that don't lend themselves to other forms of management as much. And the final thing is we now have methods for transferring agents between sites that have proved effective. And so that means that we're no longer kind of limited by the number of insects that we can rear in captivity each year. We still have limitations in terms of labor and how long things take and travel, but those are things that can be kind of remedied much more easily. And we do see a situation now where we can increase quite dramatically the number of sites that we release in each year. So I want to say a quick thank you to, again, all of our collaborators and many of the people that have worked with us. There's too many to kind of mention in one go. And as I said, this is a program or a project that's been going on since the late 90s, if not earlier. We're in a very fortunate position in Ontario to be the ones actually implementing it. But there have been a huge number of people that have put the project where it is right now. But since 2019, when the insects have been in Ontario, no one's done more really than Ducks Unlimited Canada and Nature Conservancy Canada in terms of supporting us and making it possible for us to get the work done. So a big thanks to them and to all of you for listening. And hopefully I haven't gone on too long, especially with the fish finger break. And I'll answer any questions you guys have.
Well, thank you very, very much for this. Oh, way to go. For this wonderful talk. It certainly opens up vistas to us. We have a couple of complicated sites that don't lend themselves to any kind of chemical control, and it might possibly be the solution to try, say, on some of our shorelines. It would be really great. So anyway, from my point of view, I'm wondering if you are still open for sites.
Well, so I think in terms of our 2025 releases, so we have the the kind of position we've taken for this current year. In 2024, we released across 20 different sites, which was a very good thing to kind of get the insects spread far and wide and get populations started. But just kind of anecdotally, what we find is that if we conduct small releases, the insects do well and the populations rise, but they rise kind of very slowly year on year, and it's going to take a long time before you get to a stage where a whole large Phragmites patch is kind of inundated or has a a level population of these agents. Whereas some of the sites where we've conducted really large releases for kind of experimental purposes, maybe within 2 or 3 years, those sites are kind of almost every stem you look at has damage from the agents, and you start to see these impacts like reductions in panicles on a site level. And so what we want to do really is, because we now know that we can transfer insects from nurse sites into new locations, we want to focus on generating as many of those nurse sites as we can. And the plan for 2025 is to kind of focus in the Toronto, Durham region area, and to kind of have 5 or 6 really large releases in the hope that we can generate those nurse sites that then in future years can be used to kind of inoculate hundreds of sites around that region. And so we don't currently have moths available for releases in 2025, but that doesn't mean to say that they won't become available, because certainly towards the end of the field season, we get to a stage where there are some moths that are still hatching that haven't been kind of designated for anywhere in particular. So if you find yourselves in a position where you're interested in a biological control release, do just kind of keep us updated, because you'll be at the very top of the list for any of those extras. And then certainly in subsequent years, potentially after some of the kind of conventional control that's being done at Lake Scugog, I think that might be a kind of a better time for us to come in after that treatment and kind of release in anything that's left.
So is there a charge?
No. So there's no one's commercially rearing the insects at all. And so it's just us. So that's kind of, it's good and bad news. It's good that there's no charge, but it also means that there's always going to be more people that want the moths than we can provide them. So I don't know whether it will go in the kind of commercial direction at some point. Some of these agents do, but I haven't heard any whisperings of that yet.
Well, if I could just follow up, Ian,
Oh.
thank you so much for the presentation. I think Scugog is a prime candidate for your further efforts, and there's been years of pretty intensive mapping, and there is currently a program the township is proceeding with, and maybe Ian McDougall or Keiko or Steve from our Scugog Environmental Advisory Committee could speak to our plans going forward. But we're ripe and we're ready to go with kind of support that you might be able to bring to the program that's developing or proceeding further in the township. It would be a great complement to some of the other types of mitigative approaches we have to take. Just a question I had, you referenced some of the infrastructure issues, whether it's ditches that have drainage issues as a result of the dense growth, or sight lines, even just damage to the shoulders. But have you had any experience? You've talked about a 407 project as well. Any experience with survival from your monitoring relative to roadways, whether it's dust or salt spray or things that can, from roadway use, impact roadside vegetation? Just a question on that.
And you mean survival of the moths or survival of the? Yeah, so it's a really good question, and it's one I tried to address but not with a huge amount of success. And part of that is because although we have this new agreement with 407, which has been great, we don't have an agreement with MTO at the moment. And so many of the kind of roadside sites, and that's a kind of big frustration driving around and seeing Phragmites everywhere, but we can't release into it because we just can't get permission to work there. And so we don't have an awful lot of release sites that are exposed to that kind of disturbance. But it's a really good question. I think these, I mean, I would be guessing, but I think these moths would likely be pretty robust to it, if
Mhm.
only because they spend the vast majority of their lives inside the stems themselves. So they're very kind of sheltered from it. And then even when they're flying as adults, they're kind of, these are noctuid moths. So flying is a pretty, maybe a grandiose term for what they do. They're kind of more, they're flitting around, not far off the ground, spending a lot of time actually in the thatch, we think, kind of low down in that vegetation. And so I think they have a very good chance of surviving, but that's a very good question.
Right.
Where the places we're releasing in the 407, basically for exactly the reason that you suggest, there are concerns that, how well they'll survive in those linear patches along the road. And they also may need to be treated to some extent because those patches are more likely to kind of encroach on the road. But the 407 lands, they have several areas where kind of largely on the kind of slip roads on and off the motorway, they have huge patches of Phragmites that are kind of big blocks of the weed, and that's where we're doing our releases. So the hope is that we see kind of good establishment of the agents in those areas, which are a bit further removed from all of the road activity and the kind of the chemicals.
Right.
But then from there, we hope we'll see dispersal along those linear roadway patches. And if we don't see that, it'll go some way to telling us, how well these insects do in those conditions.
I saw a chat line question from Steve Ardron. Steve, do you want to present your question? I think you're the first question on the chat line.
Yeah, so I was just wondering if the insects have been tested on crops that aren't treated with pesticides to see if they will attack any of our crops.
Yeah, absolutely. So the kind of within the plant test list were several crop species, corn and maize among them. There would have been others that may come back to me, but there's a kind of list of 70 or so plants that the insects are kind of tested against. Those would have been the crops used because these are stem-boring insects, and so really they need grass species with hollow stems in order to feed. So anything else they're not gonna bother. But yeah, so there were all of those tests revealed kind of no even exploratory feeding on those crop plants.
But
So
what I'm saying is a lot of those crop plants are pre-treated with pesticides before planting.
oh yeah.
So if, say, in a world that pesticides aren't used on crops or have been reduced significantly, will the insects be attacking plants that haven't been treated?
Yeah. So no, all of the plants that they're tested on during that kind of risk assessment phase, that part of the process was done at Cornell in New York, and they have access to various different strains of the crop plants, all of which nothing that's kind of infused with chemicals. None of that's allowed during the testing process. So the reasons why the insects won't feed on those crop plants is not to do with the kind of any kind of insecticide that they've been infused with. It's literally just the chemicals of the plants themselves. So that's what's being tested. So those chemicals on the crop plants would be another line of defense against these moths. But no, these are highly specialized feeders. And so the only plant on the test list that they showed any interest in really was the native Phragmites. But for reasons I can go into, we're not worried about that one either. But certainly the crop plants, I have complete confidence that there are no risk to those crop plants.
And just a
Great.
quick follow-up question. Have you starved the insects from the Phragmites to see if they would go after a different plant?
Good question.
Yeah, exactly. So that's, the way these plant tests work is the first set of testing. So for each plant, let's say we're testing corn, the first plant's something called a no choice test. And so in a no choice test, what we do is we take the candidate insect and we just put it on the plant and we wait to see whether it eats or it dies. So it kind of has two choices. Can it survive in any way by feeding on those plants? And most of them fail that one. Well, in fact, they all fail that one, but some of them will at least do some kind of exploratory feeding. But then once we've done that experiment, we then move on to what's called a choice test. So the choice test is where the insect is exposed to the corn again, but it's also exposed at the same time in the same arena to the target weed. So the insect is given the choice, do you go and feed on the target weed or do you go and feed on the other test plant? And so you obviously want to see that every time they're making the choice to feed on the other plant. But then we do additional tests, which are exactly what you're suggesting, which is kind of the perfect, this is what you'd imagine with biological control, is that you have an invasive weed, you introduce a biological control agent, the biological control agent controls the weed, and now you've got all these agents left that are looking for something to eat. And so that's what this test mimics, is you give them both the test weed and the other test plant. You let them feed on the test weed, so the Phragmites. But at a certain point in the experiment, you just take the Phragmites away and you determine, will any of these insects move over to feed on the other plant now? And again, none of them did. And we have kind of beyond that, those are the feeding experiments. But then we also do overposition experiments as well. So those same experiments, but with the adult moths, looking at kind of where those insects are going to put their eggs. So it could be like when an adult insect lays its eggs, what it's doing is trying to kind of provide the best chance for the offspring that are coming out of those eggs. So they're trying to look for the best quality food material for those individuals, those offspring. But some insects are better than others at kind of identifying their host plant and making sure that all the eggs end up on that plant. And so that's another form of host range testing we do, where we look at kind of, would they lay any eggs on corn in any situation? And again, they're very good at finding their host plant, and they're very host specific in that way.
Thanks, Steve and Ian. Next question is from Keiko. Have you considered species range expansion with climate change when you were testing and researching potential species interactions here, whether with other plants or hybridization with other insects?
Yeah, so I think that, in terms of the climate range, I think if anything, they will be climate would be pushing them north, which is kind of where we want them to stay, really, although people in the US would disagree because they'd like us to release them there as well, really. But I think kind of in terms of climate change, that's an interesting question. And yeah, there are models being put together in terms of what that will do to the range. But for the time being, what we're focusing on is evaluating kind of the cold tolerance of these insects to see kind of how well are they going to do surviving here in North America compared to their native range. And they seem very robust to the slightly colder winters. So I don't expect that climate change is going to do a huge amount to their range in the kind of foreseeable future.
I think, sorry, go on.
I was just forgetting what the second part of that question was.
Well, it's mostly, what happens if our, or when our winters become milder? Will they do much better here? And if that is the case, okay, so that's the one question. Would they be potentially a bit of an issue if our climate does get more hospitable here? And the other thing is when I talked or mentioned species range expansion, so you've got other plants or insects whose northern range will be also expanding our way. And potentially of some of those species, whether they're other plants or other insects, other native insects that may not be here per se, but are just, still, I guess in the region or what have you. And we do need species to expand their ranges with climate change so that we still have nature. But have you looked at potential other species interaction, whether it would be hybridizing with other insects that might be trying to expand their range here, and also other plants that will try to make this place home just because of natural selection and range expansion and weather changes?
Yeah, yeah. I mean, yeah, it's a very good question. In terms of range expansion, I think the limitation that these moths would have, because they're a single generation species with an overwintering egg, is that the temperatures could reach a level where the eggs can't overwinter effectively. And so what the kind of effect of climate change, if there is one in the kind of coming years, which there will be, but whether it's big enough to kind of to have a noticeable impact on the range of these insects, the effect would be to kind of to push them further north somewhat and limit their range. So I'm not worried about kind of extensive, range expansion beyond where we want them with these insects. In terms of hybridization with other insects, that's another great question. So you can't, you can never, and this is, some insects, even highly specialized herbivores like these, they can genetically change in such a way that they can become able to feed on another species. It's possible, but it's no more likely, for example, for these two species that we're releasing than it is for any number of native herbivores that are already here to kind of expand their range to feed on something different. And so I think it's one of those things where, when we decide about biological control, there are it's about risk mitigation but understanding the risks. Certainly hybridization with other insects, I don't think that's very likely, because there's nothing apart from the two insects that we've released which don't appear to hybridize. There's nothing kind of so closely related that I would expect that they would hybridize. But again, there's never any guarantee of that. That's what we would imagine would happen. In terms of kind of justifying the risks of those things, if you looking at the kind of native Phragmites in Ontario, for example, the biggest threat to native Phragmites is the introduced Phragmites. And I think on a landscape scale, if we don't do something about that introduced Phragmites, then the native Phragmites is on borrowed time, I would say. We're seeing it disappear before our eyes. And so I think for me, that's the bigger risk in this situation, would be to do nothing really.
All right. My concern with climate change also would be, is that going to exacerbate, the populations of that particular Phragmites? But Councillor McDougall, I see you pop on the screen with your hand up. Ian McDougall?
Yes, you did. Hang on, grab my video. There you
Well,
go.
there you are. Thank you. Hi, Ian.
Yes, you did. Actually, I had a question, and then some, I guess I can share in terms of what the municipality has kind of done, since I think in 2014, actually, we started talking about Phragmites and kind of, I want to say aggressively, but for a municipality of 22,000 people, I mean, and limited resources, we kind of put together as much as we could to try and tackle Phragmites. Actually, Paul Laporte spearheaded a lot of that and a lot of the mapping projects that the municipality participated in. So as a result, I think we have some really good mapping from probably about 5 or 6 years ago. I don't know that it's been updated in the last few years, but we really did have some really good mapping as to where it was, in the municipality. So we can kind of, I would estimate, project, where those pockets of growth have been since. I think we had allocated around about $10,000 a year towards Phragmites mitigation. But again, as Ian had indicated, it's really frustrating in the sense like, you know, a patch gets identified and then you discover, well, the patch is in water, or the patch is actually on a region property, or the patch is partially on a private landowner's property, or on an MTO property. So then you're stuck with, when you're looking at patches that you're trying to eradicate or work on, you're limited to ones that are strictly on the municipality's lands and strictly on land that's dry, generally speaking, if you're using some kind of herbicide. I think we did try the spading method one summer and some cutting, and that was rather difficult because we were using some student labor, which requires training in terms of using sharps and you know, power tools to cut things, and they need to work together with people. It's a, you know, so this is rather exciting for me in the sense that my question I typed on here was, you know, are we expecting exponential growth from these these agents, you know, moss? Like, you know, you suggested that they lay about 150 eggs a year. You've dispersed about 30,000. I know you lose some, but let's just negate that for now. So we're looking at 30,000 out there now. In a year from now, if they all lay 150 eggs and they all survive, we're looking at 4.5 million moths next year. And then the year after, it goes up to, you know, 675 million. I mean, I know we're all familiar with exponential growth, but is that what you're hoping for? And that the idea is in 10 years from now, with the purple loosestrife that you gave the example of, that this, even if we have an expanded range of an invasive Phragmites population, it's a much weaker, has a much weaker hold on its on a whole.
Yeah, I think the second part of that is bang on in terms of that's what we're hoping for. In terms of the moth population growth, I wish it worked in that exponential way, or maybe I don't. That sounds a bit scary, but there's, I mean, what we tend to find in our release sites is pretty modest increases in the population each year. And certainly kind of if we, a lot of it's to do with kind of propagule pressure. If we release huge numbers of insects, then those kind of differences are bigger each year. And we can get to a point where, you know, we have some patches that are kind of, let's say a patch the size of a hockey rink or something. And within 3 or 4 years, you get to a stage where it's very obvious throughout that whole patch that there's damage. And what that does is it means that kind of when all the adults emerge that year to fly, they're all looking for Phragmites stems to lay their eggs on. And they get to a point where they can't really find them. And so then they kind of disperse off into new locations. And that's when we get kind of new Phragmites infestations or pockets of Phragmites being inoculated with these moths. And so I see it kind of, but that new patch of Phragmites may only have a few moths in it. So it's going to take several years to build up a population in that one that's big enough that they're dispersing from there. And so, you know, this is happening from many, many pockets of Phragmites, but I expect it will take a long time. We're probably talking kind of 10 years before we get to a stage where you could look at any Phragmites in southern Ontario and reasonably expect that there might be some moths in there. And that's where we are with the kind of the purple loosestrife biological control agents. If you go into a patch of purple loosestrife, you'll find those biological control agents, but that's taken kind of 20 years to get to that level. I think in some ways there's a frustration to that, but it's also kind of why biological control works, and it works because it's slow. You know, some of the other control methods, as brilliant as they are, you know, if you need to kind of clear a roadside so people can see around the corner, you need herbicides because that'll kill things in a couple of weeks and then it's gone. Biological control won't do that. But similarly with herbicides, if you spray a patch of Phragmites, what you're left with is just nothing. And then into that nothing comes very often more Phragmites or other invasive species. And so there tends to need to be a kind of active restoration activity in conjunction with those methods. Whereas biological control works so slowly that it just kind of weakens the Phragmites enough and slowly enough that some native stuff and stuff that's in the seed bank and may have been there for decades just starts poking its head up, and you see these kind of improvements in the ecosystems.
Do you have a follow-up, Ian?
I was just gonna say I kind of wish you had a cost recovery control method for I think I think as the environmental committee we would probably, I'm guessing, I can't speak on behalf of everybody, but rather spend some of our funds towards looking for something that would work in the longer term and might be a little better for the environment than herbicides might be in the long run. But that's the future perhaps. And then my other question was, I don't know if I answered the questions in terms of what the municipality is doing. I did want to say that I know that the group had reached out to the director of public works, and he provided me with a message that said we have $5,000 allocated towards Phragmites this year. We usually have $10,000 that was in our environmental committee's budget, and it's been moved over to capital, and that is being used in partnership with Huerta Conservation for a Japanese knotweed program that's been started on the Williams Creek. So I hope that helps.
Good. Thanks, Ian. Mike Brock, did you want to present your question? Or I can read it. Mike, you there?
Yeah, I'm here. Yeah. I was just wondering how far we are from having nursing sites that have strong populations of the biological control agent, such that we could possibly harvest 100 stems, several bundles of 100 stems, and then pass them out to people in the community to introduce to their own patches near their homes or in their communities.
Yeah, no, it's a great question, and I think kind of there needs to be some thought in terms of what's the best way to do that? What's the best way to kind of like pass the labor of the biological control program and spread it across like the community? There are probably issues that I'm not thinking about, like insurance and things like that in that sense. But the first thing is, as you say, to kind of generate those nurse sites where such a thing would be possible. And those nurse sites exist already, but only kind of dotted very kind of sporadically around the landscape. So that's why kind of we're hoping to kind of focus this year on like one Phragmites management area, which would be Toronto and Durham region. And then within that region, try to generate 5 or 6 of those nurse sites. So once you have a region where you're kind of, within that project management region, we have CLOCA, that's the Central Lake Ontario Conservation Authority. And they've been running a kind of really effective kind of public group where it kind of brings together all of the people involved in Phragmites removal. And so having that level of communication within the project management area and knowing where these different nurse sites are, those are the kind of steps that we need to reach before we can get to a stage where what you're describing can happen. Because I think through that, people can get in touch with their local conservation authority or even Ducks Unlimited Canada or University of Toronto, and they'll be directed to this information that will tell them sort of not only where these nurse sites might be, but how to transfer those insects from one site to another. I think in the foreseeable future, we're looking to just continue to do those releases ourselves, possibly hire some extra staff to help us do that. But I do see in the distant future, probably it may be 10 years from now, but I do see a situation where just members of the public can kind of find out where the moths are and move them themselves.
Well, that'd be great. Okay, I think we'll conclude very soon, but one final question I'll pose on behalf of Steve Ardren. Do we have any native species that will predate on the moths and larvae that you're aware of, Ian?
Well, so very few. We're not aware of any arthropod species that feed on them, and that's in part because they're kind of in the Phragmites stems for such a large proportion of their life. So that's not, in terms of kind of shifts in the food web, that's not something I'm worried about. There is some bird predation. We do see the occasional stem that's been pecked at by a bird. So I think they'll get the odd caterpillar, but not enough to kind of shift their basic diet.
Okay, excellent. So it's just past 8:30. That was sort of our window, and thank you everyone for your participation tonight. And there's so much we could cover. on this topic. I had a bunch of questions about spraying and the other types of mitigation that you've obviously spent a lot of time considering as well, Ian. But on just behalf of our group, thank you so much for a tremendous presentation, and what an interesting, I'd say, new opportunity, dealing with this invasive. This is really exciting stuff. And to Ian McDougall's point, if we can look at exponential rearing of these insects, we've got something in hand. I just want to let everybody know our next science social will be May the 20th. It'll be in person. We have a couple of topics in mind. We'll let you know in due course, but May the 20th is our next session. It'll probably be at the Church of the Ascension, as we've held our previous ones. So please get that in your calendar. So on behalf of the Scugog Lake Stewards, we had a number of our board members present tonight. Thank you, Ian, and just thanks to everyone who participated. Hope you all found that really, I think, quite motivating. So thank you, everyone.